Hello everyone, my name is Mohit. Thank you for joining me. Today I'll be talking about an introduction to systems biology and how some of these approaches which are commonly used in systems biology can be used to better understand the dynamics of progression of cancer. What exactly is systems biology? Let me begin with that. Over the past few years, biology has been largely descriptive in nature, which means that we have identified different components and different parts that contribute to the functioning of different biological systems at varying scales, both in length as well as that of time. What has happened around the turn of the century is that all those different components, those different aspects that have been collected, people have started integrating them and trying to understand biology as an overall system, which means they don't necessarily look at the function of a particular component, but the function that can emerge, the properties that can emerge when all these components are put together in a specific format. Let me try to explain that argument with another example. Here, we see an engineered system. This particular system, airplane, has thousands of computers or chips, various different feedback loops that have been built into this. And please remember that this particular system was designed by us, the Homo sapiens, the humans. Therefore, we know the design manual which is available for this. And this was designed largely by following a bottom-up approach. What does that mean? Is that engineers first came up with smaller circuits, smaller components which would fulfill a specific function required for the overall functioning of the system, and then put them together in a specific format so as to achieve the overall objective, which is to fly passengers from one place to another. Now, compare that to a particular biological system. Here shown is a network from bacterium E. coli. Now, bacterium is one of the simplest biological organisms, as some of you would like to say that. Even in that simplest organism, there are many more feedback loops than that complicated engineering system that I just talked about. And please remember that this E. coli, this system was not designed by us. In fact, this particular biological organism might have faced different bottlenecks during its evolution and have adapted to perform certain functions in specific manner. And where does those functions emerge from? They emerge from the interconnections between these different molecules, each of which is basically a microcosm or which plays a specific subpart in performing an overall function. So the question that systems biology try to ask is, can we understand and when required, for instance in disease situations, fix such biological systems? Do we know why are the biological systems designed the specific way they are? So far we have been identifying how are these biological systems designed. Now once we have the map of various of these interactions, the questions we are trying to ask is, why are these maps the way they are? What properties do they provide to specific biological functions or organisms in this particular context? So therefore, as you may have realized, systems biology is like solving an inverse problem. Now, engineers typically solve a forward problem. What is a forward problem? Is that you are given a specific objective to achieve, and then you have to come up with a network design, a system design, which will achieve that particular objective. For instance, think of the thermostat in your room. The objective of the system is to maintain or particularly attain a specific temperature that has been set. How does it do that? It first measures the temperature, that is the ambient temperature, and then calculates the difference in the set temperature and the ambient temperature, depending on which it either cools or heats the room, after which it continues to take the ambient temperature reading at multiple time points, and at each time point it actually compares with whatever the temperature was set, and once the difference between the ambient temperature and the set temperature is minimal, it basically the temperature is achieved. So again, the objective is to attain a specific temperature, and different engineering components have gone behind the design of the system to achieve that objective. In systems biology, what we have is the function of a specific organism and or a specific cell or a specific tissue. What we do not necessarily have is the answer to how these systems were designed. So we have the different components, we have a good understanding of some of their interconnections, and we have some understanding of the function of these different organisms or these different biological systems. So what we are trying to do is to solve this inverse problem, to ask the question again, which I was mentioning on the previous slide: why are systems designed the way they are? What are the properties of these systems in terms of engineering aspects, such as, for instance, robustness of a given system? Therefore, systems biology tries to identify the design principles of these systems which exist in nature and the systems which are not designed a priori by engineers, for instance, as airplane is designed by engineers. Therefore, systems biology, or this entire approach, actually sits at the interface of science and engineering. Now, the question arises, do we actually need a systems approach to understand biology? As I mentioned earlier, biological systems function across many length scales as well as many time scales, from protein interactions, which are happening at nanometer scales, to cellular interactions, to tissue level interactions, to formation of different organs, and then finally a human organism. Therefore, it spans multiple different length scales. Similarly, in terms of the time scale of these interactions or time scale of changes in the architecture or regulation of any of these scales, it again varies anywhere from nanoseconds in terms of years. Each of these scales, both in length and time, is contributing in some complicated and complex manner to the functioning of the overall system. The other aspect is that biological systems are often nonlinear. For instance, if you know that by investing $100 you would get $120 in return, then you can multiply that and say that if you invest $10,000 you will get $12,000 in return. This is what is called a linear system, where you can linearly extrapolate the outcome of that given system based on the input that is being given. Now, what is a nonlinear system? So again, if you have a plant and then you offer to it 1 tablespoon of fertilizers, and let's see that you get 50 flowers that blossom in a specific period of time. If you give 10 times the amount of fertilizer, do you expect that the number of flowers to be blossom would increase 10 times? Of course not. The plant will eventually die because of overdose of this fertilizer. So again, this is a non-linear system, because once you multiply the input, you don't get the output in the same ratio, rather you get a completely different output. Similarly, think of a case. If you forget to take a drug or a medicine and a particular day which has been prescribed daily to you by the physician, you don't necessarily take it twice or multiple times the next day to try to compensate for that. That's not the best strategy to do. Why? Because biological systems are actually nonlinear in nature. Now, is this nonlinear dynamics important at all? So we have been talking about the example of a plant, which is the entire organism, on the previous slide. Now let me shift gears and try to talk to you what happens in a cell. So think of a cell here in which this particular protein, which has been very well studied, called P53, this is a major protein involved in how cells respond when the DNA is damaged. Now, as you see here, there are 3 different dynamics of P53 which is possible in a given cell. And those different dynamics actually determine what happens to a cell once its DNA is damaged. So once the DNA damage is being sensed by the cell and the levels of P53 are upregulated, as seen in all 3 different cases initially, then either those levels can just continue to stay high, as you see in the last case, which eventually leads to the phenotype called senescence, or it can continue to oscillate, in which the cell dies, which is called apoptosis, or it can oscillate once, which is that the levels increase and then settle down to close to what it were earlier before the DNA damage signal was sensed. In that particular case, the cell is actually able to repair the DNA, arrest the cell cycle, which means that it doesn't divide immediately until the DNA is repaired, and then carry on with its regular functions. So as you see, in all these three different cases, the input is the same, but cells are responding differently. Where are these different dynamics coming from depends on the dynamics of the networks which are present in each of these cells, which may be slightly different than one another. Let me give you another example at the level of an organ now. So here is a skeletal muscle. What happens if in one particular case, this skeletal muscle faces cycles of hypoxia? Hypoxia is lack of oxygen. So once you're exercising, then what happens is that the tissue begins to face lack of oxygen, lack of continuous supply of oxygen. In that particular case, actually the protein synthesis and growth hormone increases. However, if the system is facing or the tissue is facing chronic hypoxia, which means that the lack of oxygen is continued for a very long period of time, then that response is absolutely different, and instead of increase in protein synthesis, what you observe is increase in protein degradation. So again, these systems, these tissues are sensing for how long is that particular signal given, is that stress present, and then depending on that, coming up with different adaptation strategies. This is what is giving rise to the nonlinear dynamics which is going on. So how do we understand such complex systems as I just talked about? Let me give you another example: weather prediction. Today we are at a stage where we actually trust these predictions which come up on our television or now on our smartphones. But of course, the stage which we have reached today is actually the hard work put in by various scientists, be it climate scientists or mathematical modeling people, physicists, in trying to understand what are the different parameters that determine the weather of a particular place at a particular point of time. And then how do those different parameters, those different variables, how do they crosstalk with each other? Are there general principles, general equations that one can write down and therefore try to predict the outcome? So of course, this has gone over various iterations where mathematical models have been made and then matched up or validated against the data that was available. Further data has been collected, the models have been improved, and in this iterative cycle, we have arrived at a stage today where we can actually relatively accurately predict the weather forecast or at a given place at a given point of time. So can we reach a similar level of predictive and quantitative understanding of the functioning of different biological systems is the question that systems biologists try to ask. Now, the word understand may have different meaning to different people. If you speak to most experimental biologists, one of the meanings of the word understand that you would get to know is that different components of that system have been identified and they have been characterized at molecular level and possibly at a functional level. However, if you speak to engineers or physicists, their meaning of the word understand is slightly different. For instance, Richard Feynman famously said, "What I cannot create, I do not understand." So in terms of engineering disciplines, the meaning of the word understand is that one is able to make accurate predictions, quantitative predictions about the functioning of a particular system given a specific input, which is is one of the aims of synthetic biology, that is constructing biological circuits which will achieve a specific objective, similar to the thermostat that I talked about. Now, synthetic biology, as we know today, there are two landmark papers that appeared in the journal Nature in 2000, which actually spearheaded much of the developments that we hear about today. In these two papers, the authors actually constructed biological circuits of a specific topology, which we'll go into details of later in the webinar. And then they were able to predict how these systems would behave. And finally, they experimentally showed by inserting these systems again, in E. coli and showing that this is how these particular systems behaved. Therefore, subscribing to Richard Feynman's definition that they were able to create these systems, insert them in biological scenarios, and then actually show this is how these systems were behaving. So they gained a complete understanding of the dynamics of these specific cases. Now, synthetic biology has come a long way. We can actually see the possible applications of synthetic biology in various different cases, not only in E. coli as we talked about on the previous slide, but also actually in different metabolic engineering aspects such as increasing the biofuel production of a specific molecule or enzyme, or also in delivering drugs in a very periodic manner, or coming up with new therapies such as cancer immunotherapy, which actually won the Nobel Prize a couple of years ago. Now, one of the aspects that I've been referring to in the context of systems biology is that of mathematical modeling. The question that arises is that why should we mathematical model a particular biological system? What new insight can we gain by following this particular thing? Before we go into those details, let us think about what exactly is a model. A model is a replica of the original system that we are trying to build, which is used to predict how the original system will behave. For instance, when engineers try to design the airplane that I talked to you about, they actually make different models on and then of the airplane and then try to understand how this particular model, this cartoon, this schematic will actually behave if you vary, say, the air pressure. And based on that, they actually continue to change and evolve the design of this system so as to achieve the specific objective. Similarly, in the context of biology, experimental biologists often use a model organism. What is a model organism? As Wikipedia states, it is a non-human species that is used to study in the laboratory different biological phenomena which are of relevance to human biology, so that with the hope that if we understand that particular phenomena in the model organism, one can apply those insights to understand how exactly the human biological system is operating. As the famous statistician George said, essentially all models are wrong, but some are useful. So therefore, whether it's a biological model or a mathematical model, each comes with its own assumptions, strengths, and limitations. Therefore, we should take whatever we learn in a particular model, be it a mathematical model or a model organism, always with this pinch of salt that this has been studied in these situations and may not necessarily directly translate to our understanding of human biological systems. So what exactly a systems approach helps us is that it helps us to explain how a system works and why it is designed the way it is. It can also be used to predict the behavior of that particular system under some novel conditions which that system may face, and eventually, as I talked about, to engineer a system, to create a system, to construct a system to achieve certain design outcomes. Therefore, the field of systems biology actually borrows tools both from physics and mathematics in terms of identifying those design principles of specific biological systems, as well as from engineering and systems theory in terms of actually constructing circuits which can eventually be used to achieve specific outcomes. And of course, all this information is coming from different biological systems. Therefore, as I said earlier, systems biology or this approach actually sits at the fence of science and engineering. Now, I must emphasize that despite the complexity that has been observed in biological systems, they actually still function. Complexity does not mean that they don't know how to function. In fact, biological systems function in a very robust manner. If you think of the process of development of an embryo of any organism, it is actually a fantastically robust process. If you think of the different levels of organization that need to be coordinated both in space and time. So how do we get this robustness, this reproducibility in the functioning and the organization of biological systems despite such complexity? It happens because what people think that there are some rules, there are some design principles, there are some set paradigms which emerge as a result of interactions happening between these different systems on how can specific things be organized. So biological systems therefore are actually very tightly controlled, very tightly regulated at different levels, both within a cell as well as across cells in a tissue and so on, so as to give that particular achieved desired outcome that we have been talking about. I talked initially about feedback loop. And feedback loops, as you may have realized, is actually a very important component of designing a particular system to achieve a specific objective. So here is just an example of an customer satisfaction feedback loop. So these companies go out and ask the customers if they like the new product, and then depending on the feedback that is received, the new information that they have collected, they actually fix the product, improve it, make changes, and then bring the updated product out in the market, and then check whether the sales went up or down, and continue to collect this feedback from the customers. So therefore, this information that is received from the customers is actually helping them optimize the design and achieve the desired outcome, which in this particular case is to maximize their sales. So what are the general components of any feedback loop? Feedback loop receives inputs, it has some outputs, and then based on that output, the input can actually be varied, which is the very general principle of any given feedback loop. So what we'll do now is actually study different biological systems in which similar feedback loops are actually known to be present. So again, what is a feedback loop? That in response to a stimulus, there is some sensor which integrates all the information coming from the outside, based on which the cell particular response in a specific way, which is called the control or the effector, and then the information based on the decision that the cells have taken based on the information again goes back and continues to alter the dynamics. For instance, let's talk about our body temperature. So we all know that our body temperature is maintained around 98 degrees Fahrenheit or 37 degrees Celsius. Once the body temperature exceeds, there are mechanisms in the body to bring it back. For instance, the nerve cells in the skin and the brain, when they receive this information through the nervous system that the body temperature has increased, what you actually see is that you start to sweat. Why does this sweat happen? Because the central nervous system, the temperature regulatory unit which is present in the brain, actually conveys information to sweat glands to secrete sweat, because that will help reduce the body temperature back to normal, which is around 37 degrees Celsius. So this, as you can see, is a negative feedback loop, where what this feedback loop is trying to do is actually to go back to where it was set, or what the original condition was before this particular perturbation happened. Similarly, body temperature falls, tend to shiver, and this process of shivering actually generates more heat, therefore increasing the body temperature. Again, in this particular case, the objective of this entire system is to bring the body temperature back to normal in a specific predefined range, which is called homeostasis, that is the process of maintaining a relatively stable internal environment. Body temperature is one of our vital signs, and it is important to maintain it in a specific regime so as the body can function properly for various different objectives. There's another feedback loop in our body which actually regulates blood sugar. Again, that's an important vital sign. And there is a homeostatic level of blood glucose in our body. Once that level either increases or decreases, there are mechanisms to bring it back close to where it was initially, which is 0.1%. Once the blood sugar increases, messages are sent to the liver to begin to store glucose as glycogen and bring it back. Once the blood sugar decreases, a different message is sent to the liver, which is to convert the glycogen that was stored earlier to glucose. So therefore, the storage of glycogen, as you can see, basically acts as a backup mechanism in case the blood sugar decreases. There is another feedback loop, which is trying to maintain the levels of calcium in our body. Again, if there is an increase in the calcium level, then the thyroid gland actually releases this particular molecule called calcitonin, which helps restore the levels of calcium to whatever normal levels of calcium are supposed to be in the body. Similarly, when there is a decrease in calcium level, parathyroid glands release another molecule which is important to bring back the calcium level again to what the normal set homeostatic condition is. So as you saw in three different cases, the design principle of these feedback loops is exactly the same. Their objective is exactly the same, which is to maintain homeostasis, and therefore whether when there is an increase, this particular feedback loop tries to bring the levels of that entity down, and when there is a decrease, this particular feedback loop operates in order to increase the levels of that particular aspect to bring it back to what the homeostatic condition is. So this imbalance that may be created due to various different conditions in our body or various different conditions outside our body, such as the environment. In that particular case, there are different mechanisms which can be mapped onto the idea that we talked about, that of a sensor control system, center and effector, whose overall goal is to bring it back and make sure that this balance is minimized, the imbalance is minimized and the balance is restored. There are other kinds of feedback loop which actually do not reset the system to where it was initially, but actually tries to amplify the response so that the system can reach a completely different state. For instance, in the context of the delivery of a baby during childbirth, what happens is that once the head of the baby starts to push against the cervix, there are different biochemical messages which are sent across the body of the mother, and what that entire biochemical message pathway tries to do is to push the baby towards cervix. So again, the initial stimulus which is generated by the baby to try to push the cervix, that stimulus is getting constantly amplified through these different biochemical mechanisms. And what happens as a result is that once this amplification is strong enough, then the baby is delivered. So here we actually see that the system does not come back to where it was. The system does not push the baby back in, but actually leads to the delivery of the child, leads to childbirth. Therefore, now there are two states of the system. One in which the child is still inside the womb, and other in which the baby has been delivered, which is the amplified response. So these are positive feedback loop, which can lead to these two distinct states. Another positive feedback loop is observed in the context of blood clotting. For instance, when there is a breakage in the vessel wall, what happens is that clotting begins, and the molecules which are secreted by the cells which are responsible for clotting basically trigger another whole set of events which lead to an increase in platelets which are actually contributing to clotting. So again, what you see is an amplification of a response, or again two states, one in which there is a break in the vessel wall, and another in which this break is actually sealed by platelets which are beginning to clot. Now, of course, this feedback loop has to end somewhere. So just in the case of childbirth, once the baby has been delivered, there is no more stimulus which is going from the baby to the cervix. Similarly, once the wound has been healed, then the particular molecule which is secreted to actually recruit more platelets is dampened and eventually dies out. Therefore, this particular feedback loop gets broken. So, positive feedback loops and negative feedback loops have very different functions in regulating different responses of our body. So far, we have talked about these operations happening at an entire organism level. Let's now talk about some feedback loops which are operating within a single cell. So for instance, P53, the molecule that I introduced to you earlier, actually regulates itself via a negative feedback loop. P53 increases the levels of another molecule called MGM2 in the cell, and this particular molecule is able to degrade the protein P53 in a cell. Therefore, this forms a negative feedback loop. And this negative feedback loop can, in some cases, give rise to these kinds of oscillations that you observe. Now, what happens in cancer is that P53 actually is one of the most commonly mutated gene in cancer. What that means is that in many cancer patients, there are changes happening to the structure and or function of P53, which basically disrupts this feedback loop. And once this feedback loop is disrupted, this mechanism of homeostasis at a cellular level is disrupted, which actually may contribute to formation of cancer. What is cancer? Cancer is basically uncontrolled proliferation, and P53, as we discussed earlier, is one of the molecules which restricts cell cycle, which restricts abundant proliferation of these cells. Now, once this molecule is disrupted, once this molecule is perturbed, due to different mutations, this feedback loop is broken. Therefore, the function gets compromised and the cells begin to proliferate at a much faster rate and with much more intensity as compared to normal cells. Therefore, these negative feedback loops, which contribute to homeostasis, once broken, can actually lead to diseased situations. Let me give you another example. When cells are continually dividing in our body, there is a mechanism called contact inhibition, which basically makes sure that the number of cells at any given point in the body do not exceed a specific limit, a specific threshold. So when these cells come in direct physical contact with each other, there are some biochemical molecules which are exchanged, which makes sure, or it tells the other cells that, look, there are enough of us around, so please do not proliferate, please do not grow. Again, what happens in the context of cancer is that these biochemical molecules actually get compromised. And that again leads to a continued proliferation or a much higher number of cells dividing and growing in the same amount of space, therefore disrupting the architecture of that particular organ and eventually contributing to a loss of function of that particular organ, which is what seems to happen in the context of cancer. Now, the questions that arises is, do cancer cells always use broken feedback loops, or they actually have negative feedback loops of their own which help them adapt? Now, think from the perspective of a cancer cell. Its environment is constantly changing, and what negative feedback loops help do, as we studied previously, is that it helps the system to come back to a particular homeostatic state and therefore adapt to different perturbations that the system may have faced in that period of time. Therefore, a function of a negative feedback loop is to adapt the cells. So it is possible that cancer cells may be using some of these negative feedback loops in order to adapt to changing environment around them. For instance, when they face different drugs that are being given to a specific cancer patient. So negative feedback loop has actually been talked about more recently in the context of cancer, is that it helps develop cancer cells resistance against the different therapies that are given to a specific patient, including one of the studies that we were a part of, the context of ovarian cancer, where we observed that when these ovarian cells are developing in the very initial phases resistance against this particular therapy, they actually make use of a negative feedback loop. And once they have established that particular mechanism, that feedback loop is no longer very frequently used. So the dynamics of the evolution of resistance also involves these different negative feedback loops across different cancers and possibly across different drugs as well. Now, are there positive feedback loops which are operating in the context of cancer? Yes, because again, if you remember, the function of a positive feedback loop is to take the system from one state to another. Now, think from the perspective of a cancer cell which has left its home organ, what is called primary tumor, which is where the cancer originates. And now via bloodstream, it has gone to a different organ and it needs to settle down there. The conditions in that particular organ may not be very conducive both at biochemical level or at biomechanical level to the growth of this single cell or a cluster of cells that have reached a specific case, a specific organ. So what these cells, these cancer cells need to do is to remodel the environment around themselves at that foreign location so that the new environment is actually conducive to their growth. And examples of this has been seen in the context of prostate cancer. Once these cancer cells actually reach the bone, which is the most frequent observed metastatic location for prostate cancer. Once these cancer cells reach there, they're able to secrete different enzymes, different molecules that are able to change the behavior of the cells which are already present in the bone in a way that they now begin to support the growth of cancer cells as compared to the previous scenario where they were actually not very supportive. So they have this ability to hijack these cells and use them to their own advantage, where again, as you can see, different kinds of positive feedback loops which can amplify such a response are present. So we have already talked about two of the very simple feedback loops. One in which two aspects amplify each other, one in which there is a negative feedback loop which leads to a homeostatic response. Now, if you map them onto what is going on within a cell, you can actually represent them as the following. So if you consider two proteins that are changing the levels of one another, in one case they are increasing the levels of one another. In another case, what you see is that one of the proteins increases the levels of the other, whereas the other decreases the levels of the initial protein that upregulated itself. For instance, in the case of P53 and MDM2. So there's a double positive feedback loop, or it's a positive feedback loop which leads to amplification, and then there's an overall negative feedback loop, or you can call it a negative positive feedback loop, which actually leads to a homeostatic response. The questions that, again, systems biology tries to answer is that how does the system work and why is the system designed the way it is. Now let me talk to you about a different kind of feedback loop, which is that when both of these proteins actually inhibit each other. So you can think of this scenario as two people who hate each other and do not want to go to a specific party. So if A is present, it would restrict the presence of B, and if B is present, it would restrict the presence of A. So as you can now intuitively think about the system, that this kind of system may lead to two different outcomes. One in which the levels of A are high, or in other words, A is present at the party and the levels of B are low, which means B is absent from the party. So A high, B low, or A present, B absent. The other possible scenario is that when the levels of B are high and consequently the levels of A are low, or again if B is present at the party and A is absent. So this kind of feedback loop can actually lead to two different responses, and therefore it is often called a bistable system. Bi means two. The two responses being A high, B low, and A low, B high. Now, where are these kinds of systems, these kinds of feedback loop seen in biology? So often in a stem cell, which are special kinds of cells in our body which have the ability to give rise to multiple different types of cells in our body. When a stem cell has to decide which cell fate should it adopt, that is when such feedback loops become very important. Because imagine just a hypothetical scenario that there is a stem cell in the body which during the process of development has to decide whether it should become a lung cell or it should become a muscle cell. So A and B, these two different molecules that we are talking about, consider them as some master regulators of being a lung cell or master regulators of being a muscle cell. Now, if that cell has to become a lung cell, its muscle identity needs to be switched off. And similarly, if it has to become a muscle cell, its lung identity needs to be switched off. So what needs to happen is an exclusive commitment to one of these two fates in which only one of these molecules is high and the other molecule is actually low. As I was mentioning, this kind of feedback loop are seen at different stages of development of an organism where these different decisions have to be made in terms of what are called as sister cell fates or two cells which can come from the same progenitor. So I'm just showing you examples here. These two molecules A and B, or in this particular case called X1 and X2, you can see that at different branching points from the point of view of development, different molecules, each of which is a master regulator of one versus the other cell fate, actually regulate each other in the same way. So again, there is a design principle to it. The reason why these feedback loops are present at the places where they have been experimentally identified is because you need exclusive commitment to one cell fate at that decision point. Now again, that is in the context of development. Do cancer cells also use such toggle switches where they can switch back and forth between two different outcomes? One in which the levels of A is very high and B is low, other in which the levels of B is very high and the levels of A are quite low. So let me tell you a little bit more about the process of cancer metastasis, and then we'll discuss why some of these toggle switches are actually seen in the context of cancer, particularly in the process of metastasis. So what is metastasis? As we talked about, it is the process of cancer cells from going from one organ to another, and it still remains the cause of more than 90% of all cancer death. Now, how do cancer cells metastasize? Is through this fantastic set of freeways which is present in our body called the blood vessels. So imagine cancer cells which get on the freeway from a specific organ, and then they're able to actually traverse to all different parts in the body and then take exits to actually get out at distant organs in the body and begin to form tumors there, which is the process of metastasis. Now, if you look at the most common cancers that you may have heard of, say breast cancer, prostate cancer, lung cancer, liver cancer, etc., those cells do not have the intrinsic property typically to begin to move, to begin to invade. Those cells actually are set in a specific 3D geometry, are very tightly adherent to their neighbors. So how are they able to change this state, which is now from a tightly adherent stationary state, they're actually able to go to a migratory phase where their adhesion to their neighbors goes down. And once they have reached a distant site, again, they are able to regain the ability to settle down and to form tumors there. So this is the switch I was talking about. And again, if you think of metastasis from the point of view of a systems, it's actually a highly dynamic process where there are constant changes that are happening in the environment of a cell, and therefore this particular cell needs to continue to adapt to it in terms of various aspects, be it its ability to adhere to the neighbors, the ability to migrate, the ability to settle down in a tumor and colonize it, or the ability to resist the different therapies that are being given. Now, again, molecular biologists, oncologists have done a fantastic job in identifying different molecules which contribute to each of these different axes that I spoke to you about. And what is the role of systems biology is to actually put these parts together in terms of how they actually interact with one another as identified experimentally, and then study what is the emergent outcome of any of such system, because then is when we'll be able to understand that how does the function, which in this particular case is migration of a cancer cell, come about as a result of these different interactions put together. So again, the philosophy of systems approach basically is that whole is greater than sum of its parts, which is called an emergent phenomenon. So again, A systems approach helps understand and eventually defeat this process of metastasis. Now, as I was mentioning earlier, the word understand may have different meaning to different people. On one hand, once we identify these different components, each of which is shown by a small green circle here and how they affect each other, that may be sufficient for some people to call it that they understand the circuit. But anyone trying to understand this from a more quantitative or predictive manner, what you see is that there is no time scale mentioned, no length scale mentioned, no information about how strong the regulation of one node to another node is, and how linear this is. Also, there is very little or no information about what happens if there are multiple links coming into the same node. Are both of them required to change the behavior of that node? Which is if you talk in terms of computer science, is it an AND gate or just one of them will be sufficient? It's an OR gate. We don't know any of those details from this particular diagram that is shown to the left. So various of these assumptions are actually hidden in a black box, and that can give rise to some unpleasant surprises in the clinic. For instance, once the anti-angiogenesis therapy was tried for cancer patients, angiogenesis is the growth of new blood vessels. So the idea was that if you stop the growth of new blood vessels, you should be able to restrict the growth of cancer. Fair idea. Once this was tried, it had to be stopped in between because some of the patients receiving this therapy were actually dying faster. And only later it was realized that one of the reasons why this was happening is because this particular therapy was actually accelerating the process of metastasis. Why? Because again, cancer is a dynamic adaptive system. You cannot think of it as a static system which would not evolve ways to bypass the different stresses that it is facing. And again, this call for understanding cancer from a more dynamic perspective, a predictive perspective, has been coming from the leading cancer biologists in the field as well. For instance, this is the last sentence of one of the most well-respected reviews written in the field of cancer biology, where they identified the different hallmarks associated with different cancers all put together. And the authors say that one day we imagine that cancer biology and treatment will become a science that has a conceptual structure and logical coherence that rivals that of chemistry or physics. Again, those are the branches of science which we were just talking about, where mathematical modeling and experiments in various different subfields actually go hand in hand. Similarly, once these same authors actually rewrote or reassessed the hallmarks of cancer, they continue to believe that these underlying organizing principles or design principles that we have been talking about still remain to be largely identified. Now, let me give a brief example from some of the work we have been doing in the context of metastasis, using the ideas that I just talked about to share how these approaches that we have been talking about can be used to understand this dynamic process of metastasis. As I was mentioning, there are these epithelial cancer cells in the body which do not have the property to migrate or invade, and then take on some of these properties of the so-called mesenchymal cells in the body, which have the property to migrate and invade, and they actually do not typically adhere to their neighbors. This transition that happens is called as epithelial to mesenchymal transition, or EMT in short. Now, this process is usually important once cancer cells begin to leave the primary tumor. Once they have reached a distant metastatic site, they actually need to regain their ability to adhere to their neighbors. They no longer need to migrate or invade, and they undergo typically a completely opposite transition called as MET, or mesenchymal to epithelial transition. So these cycles of EMT and MET that may be happening in the context of metastasis actually continues to form secondary and tertiary tumors at different organs in the body. Now, once we look at the literature of molecular characterization in terms of EMT and MET, what different molecules drive EMT, what different molecules drive MET, see how they have been experimentally reported to connect to each other, what we see is again those kinds of toggle switches. For instance, here It's a very small network, a set of molecules which have been consistently reported to regulate EMT and MET in multiple different cancers. And please don't take me wrong, EMT and MET are much more complicated than just these four molecules shown here. Here we are just talking about some of the molecules which were more commonly reported across different cancers to try to get to those design principles that we have been talking about. What you see here are these two proteins, and these tend to drive a much more mesenchymal phenotype. So if the levels of this particular protein is high, the cell usually becomes mesenchymal. On the other hand, there are these other two molecules which are trying to drive a more epithelial phenotype. So if the levels of this particular protein in these particular molecules in a cell is high, then the cells typically stay epithelial. What you see in between is this mutual inhibition, this toggle switch, this mutual hatred between A and B who do not want to go to the same party. So again, cancer cells can possibly use toggle switches to regulate their behavior once they want to leave the primary tumor and later when they want to return to a more epithelial phenotype. So now let me show you the same thing in a more graphical manner. So let's look at this graph and see how the levels of A change as B increases. So because B is inhibiting A or not letting the levels of A go higher up in the system, what you see is that as B increases, A decreases. Now in the same plot, let's look at the other way, which is how the levels of B change as the levels of A are changing. So you'll have to tilt your head a little bit to actually understand this particular graph. What you see is that this particular graph intersects at three different places. These three different intersections are places where both of these conditions, which is that A inhibits B and B inhibits A, are being satisfied. Now, what you see are in these three intersections that one of them is the point, the left top point, where the levels of A is high and the levels of B is low. Similarly, if you look at the right bottom intersection, what you see is that the levels of B is high and the levels of A are low. This is again those two states that we have been talking about, A high, B low, and A low, B high. What is the central point which is somewhere in between? Let me take you back to high school physics, where we try to understand whether a particular state is stable or not. How do we test that? That we give a little bit of perturbation. If the system returns to its original equilibrium, it's actually a stable state. But if it goes away to one of the other possible state, it's an unstable state. We actually did this analysis, and what we observed was that the middle intersection is actually an unstable state, because remember, again, between every two stable states there has to be an unstable state. So this system can behave as a bistable system, and which can, again, you can represent that through a set of equations which I'll not go into details of, but in case anyone of you is interested, I've just flashed them here. So these equations are completely symmetric. So let me walk you through one of them. What does this say? dA by dt, which means the rate at which the levels of A change as a function of time depend on is equal to GA, which is some constant production rate at which A is being produced. But again, there is an effect of B on the production of A. Therefore, the term that you see in the green box is actually multiplied. And what does that term tell you? That as the levels of B increase, the value of this fraction decreases, therefore reducing the overall production rate of A. Then the next term is minus Ka times A. What is this? This is basically a first order chemical reaction which is responsible for degradation of particular molecule of A. Every molecule has a given half-life at which it degrades. So based on that, we can actually calculate the rate at which this particular molecule is decreasing. So again, the right hand side of this equation has two parts. One is the production and the other is the degradation, and the degradation term comes with a negative. So again, this toggle switch, this double negative feedback loop, as we discussed earlier, remains one of the first synthetic biology circuits which was designed, as well as the hallmark of self-rate decision during embryonic development. Now, once we mathematically modeled the EMT MET circuit that I showed you a couple of slides ago, let me tell you what the different predictions from our mathematical model were. So one of the predictions of our model was that cells need not only maintain an epithelial or a mesenchymal state, they can actually have a third state in between, and what we called it as a hybrid epithelial mesenchymal state. The other prediction of our model was that now if you take a population of cells, because there are these three states which are possible, some cells may end up in state 1, some of them may end up in state 2, and the other of them may end up in state 3. And because there is noise in the system, and biological systems are known to be very noisy, these cells which are in state 1 at a given point of time may actually end up in state 2 after some time, and some of the cells in state 2 may switch to state 1 or state 3, which means that there's dynamic switching that is going on between these cells. So again, you cannot necessarily identify which cell is going to be in which state at what point of time, but what you can predict or estimate is how many cells are going to be in different states at a given point of time. So what is the distribution that is going to look like? So once we made these predictions, we actually worked together with different experimental groups to test these predictions. So I'm showing you just a few examples here. For instance, these are lung cancer cells where they have been stained with an epithelial and a mesenchymal protein at the single cell level. The green fluorescence is for the epithelial protein, the red one is for the mesenchymal protein. So what you observe is that most cells actually co-stain for red and green, which means they have both epithelial as well as mesenchymal characteristics, therefore indicating the possibility of this hybrid behavior. And since we said that this can be a stable state, we actually continued to grow these cells in the lab and then observed whether this particular phenomena was constantly seen, which at least in this particular cells it was. Now, further experiments were done in different cell lines belonging to breast cancer, prostate cancer, lung cancer, and colorectal cancer, where each of the cells was probed for the presence of the same molecules that I was just talking to you about. And again, what you see is that there is heterogeneity, that in each of these cell lines, which are cells taken from patients, you see that some cells have only one of the protein, some which can be vimentin, some which is shown, and that particular subsection is shown by the violet colored cells. Some have only the other protein, which is shown by green. And then what you see is that there are a few cells which can actually have both of those proteins, which are shown in the top right in each of these cases. Now, further experiments were done where these different subpopulations that I just talked to you about were segregated from one another, and then their dynamics was observed in the lab. What you see here is that again, over a period of 2 weeks, each of them is able to switch and give rise to other different states. Now, the rate of switching is of course not the same from state 1 to state 2, state 2 to state 3, and state 1 to state 3. It depends on various different features. But again, at a qualitative level, these different predictions that we made about the dynamics of the system were all validated by different experimental groups over a period of time. Other aspect that we looked at is the other feature in the context of metastasis, which is called stemness, which is the ability to grow tumors once they have reached a distant site. Now, how EMT, which is the process by which they begin to move or they accelerate to move, affects their ability to form stemness? That is the question. So one of the first reports in the field had suggested that when cancer cells undergo EMG, their stemness increases, which is shown graphically here. The left end of this axis is epithelial, the right end of this axis is mesenchymal, and the stemness window, which lies somewhere towards the right, which means that If the cells are present towards the very right, they may be present in the stemness window, and therefore they may be much more stem-like, much more tumor initiating. A couple of years later, there was a report which said the exact opposite thing, that EMT actually decreases stemness, and there was another report which said that EMT has no connection to stemness because you can have epithelial stem cells as well as you can have mesenchymal stem cells. Again, we put together different parts, different molecules regulating stemness, regulating EMT, look at how they are experimentally reported to interact with one another. Once we have a network, we again convert them into a set of equations similar to what I showed you earlier and make predictions. So what our mathematical model predicted here was that the stemness window actually lies somewhere in between, which means that the cells which are most likely to be stem-like or like tumor initiating are the ones which are hybrid cells, not the ones which are completely epithelial, not the ones which are completely mesenchymal either, but the ones which are somewhere in between. I must clarify that our model did predict that the stemness window could move left or right on this particular axis, but its most likely positioning was somewhere in between. Now, once we had this theoretical prediction, actually people started doing experiments and observed that yes, indeed, that was the case. The cells which were somewhere in between were actually forming the maximum tumors as seen in vitro experiments as well as in vivo experiments, which are experiments done in mice. That these cells which had both epithelial as well as mesenchymal characteristics were forming the fastest tumors and the maximum number of tumors. There was a paper in Nature a couple of years ago, which actually validated multiple different predictions that we have been making. First of all, it showed that there are multiple states of EMT. It's not a binary process, just epithelial or mesenchymal. You can actually have cells somewhere in between. They also showed that if you start with one of these subpopulations, which are each of them is shown by a cartoon here, and just let them grow, what you see is that they are able to give rise to other different subpopulations, which is that they are able to switch to other states because of inherent noise which is present in biological systems. The third aspect that this showed was that the ones which were most metastatic were not the ones which were completely epithelial or completely mesenchymal, but the ones which were somewhere in between. So again, these different predictions that we have been making are continued to be experimentally validated in multiple different cancers, and that is the work I wanted to present to you as an example of how some contributions from mathematical modeling of trying to understand the dynamics of these biological networks, these biological systems, can actually help us gain a better handle of how these processes of cancer metastasis are happening. Just summarizing, as we already discussed, that biological networks are complex, they are complicated, they involve various different parts interacting with one another at different scales, both in length and time. And then understanding the dynamics of these networks that are formed actually helps us understand how these cells respond to the different stimuli that they may be facing. And systems biology approaches or using these mathematical models can be a powerful tool in understanding that particular feature of these cases. Now, biological networks are complex, but embedded in that complexity are some network structures similar to what we talked about, the feedback loops, which have a specific function. And once we begin to identify those feedback loops and characterize them using experiments as well as using mathematical biology, we can get a much better appreciation of why biological systems are designed the way they are. And again, as we discussed, negative feedback loops have a function of adaptation. It is used in our normal homeostatic condition, and it can also be used by cancer cells in trying to adapt to different therapies that are being given. Similarly, positive feedback loops, they have a response of amplification during childbirth or during blood clotting, which are homeostasis conditions or physiological conditions, but cancer cells can also use these kinds of feedback loop during the process of metastasis. The other feedback loop that we talked about is this double negative or mutually inhibitory feedback loop, which enables cells to switch from one state to another and take a decision at a given point of time, seen in development, also seen in cancer. So there are principles across these different levels of organization, across these different biological setups and scenarios, which can help us gain a much more nuanced understanding of the dynamics and why the systems behave the way they do. In these webinar, specific questions, you can think about them and respond to them in the comment section at your own convenience anytime. So we discussed that blood sugar, body temperature are maintained in homeostasis via these negative feedback loops. The question is that do similar loops regulate blood pressure, because that is also a vital sign, and for the functioning of the human body it needs to be maintained in a specific regime. Are there similar feedback loops? If yes, what are the different components of those loops? Similarly, if we talk about a completely different biological setup, which is the population of different prey and predators, as have been studied by ecologists for a very long period of time, it has been shown that they give rise to some sort of oscillatory response or a pulsatile behavior as commonly observed in negative feedback loops in the biological networks that we just talked about. So if there's a negative feedback loop which is operating in the context of prey and predator, what exactly is that feedback loop? What There are two populations here, prey and predator. How are they affecting each other to form any such feedback loop? Thank you so much for your attention, and I'll be happy to have any questions which you can post as comments, and I'll come back and respond to them. Thank you once again for your time.